Welcome everyone, and thank you for joining us for today's webinar titled Monitoring Food Intake, Water Intake, and Ingestive Behavior in Group-Housed Rodents. My name is Martin Hess from Inside Scientific, and I will be your host for today's event. Our session today is sponsored by Embros and will focus on the latest technology and best practices for studying food and water intake, as well as feeding kinetics of group-housed rodents in their home cage. Our speakers today are Dr. Mas Kulbi, an assistant professor at Aarhus University in the Department of Biomedicine, and Dr. Niels Vellner, a postdoctoral research fellow also in the Department of Biomedicine at Aarhus University in Denmark. In today's webinar, our presenters will discuss methodology, best practices, and provide guidelines for implementing a real-time food and water intake protocol using group-housed rodents. Focus will be on study setup and data collection of water intake, meal size, frequency, and duration. Examples will be provided of acute intervention studies like the leptin test, as well as longer-running experiments. Methods and data will also be shown from an ethanol preference test in mice. We will also hear briefly from Mr. Renee Vestergaard of Embros, who will provide us with an overview of the Embros HM2 food and water intake measurement system so that you can get a better understanding and appreciate the information and data that will be presented by our guest speakers.
I'm Renee from Embros. I'm the sales and marketing manager here, and I've been here for approximately a year now. Okay, and thank you all for participating in this webinar. I will jump right into it. So, the introduction to the HM2 system. This is for giving you a ground-based understanding of the system, so you better understand what Mas and Niels is talking about and where they get their data from. So, I will go on. So the whole idea behind the HM2 system is based on more than 30 years of experience working with measuring behavior in rodents. For 20 years we've been working with automated food intake, and for the past 10 years we've been working with intake in group-housed animals. So we have some key factors that were set for the HM2 system. The first one is group-housed animals in their home cage environment with the individual intake monitoring. The second one was client-server solution, so we wanted to give easy access for all involved parts, also due to still rising requirements for having all animals in a controlled animal unit. The next point is an embedded computer. So we wanted to provide ample power to each station, allowing millisecond precision for all recorded events. And finally, all parts that are in contact with animals should be easy dismantled, washable, and autoclavable. So here we see the HM2 system just to describe a basic setup. So the first part is of course the HM2 station itself. The second part is the VS1 weigh station. The third part is a computer, and this is provided by customers themselves, so the computer is conforming with the local network. And finally, you see a rack system as well, and this is just to tell you that we deliver benchtop solutions, of course, but we also deliver rack solutions with a built-in rack solution. So it really depends on the scale of your research. Okay, moving on to the station itself, the HM2 station. This consists of a base station. This is for the embedded computer and display. Then we have detectors for station status. We can use standard cages on the market, and we'll customize these so they fit into the channels, which I'll come back to. We have a peer sensor, which we see on the top here of the cage. So this is an activity monitor. Important thing to understand here is that this is on the cage level. This is monitoring, so it's not on the individual animal. Okay, and the final and most important part is the channels. So we have two configurable channels with load cells and detectors. The setup can be food and water in one setup, or it could be water or feed for preference studies, for example. Okay, moving into the channels. So here we actually see the food hopper, and over here we see the water bottle. So if we're doing an alcohol preference test, it will just set 2 water bottles into the system, and then we can provide the food in the lid of the cage. Okay, before going into the technical part about the detection of meals, it's important to understand in this system that the animals need to have a microchip injected subcutaneously in the neck. So this is how we can identify the individual animal. So down here I have put in a picture of the injection tool of the microchip, and here a pile of microchips or RFID transponders. Okay, so the technical side, when an animal goes inside the channel, photocells are detecting the animals. When the detection is made, an electronic identification is done by an antenna, and due to load cell instability, a meal event is registered. Another key part in this is, if you notice down under the feeding channel and water bottle, we have a tray. This tray picks up spillage and therefore deducted. So this is deducted. So a precise intake of the animals. Okay, moving on. Another key part of the system is the VS1 Weighin Station. This station is a combination of a scale and an antenna for electronic identification. So when you place an animal on the scale, the microchip is read, the body weight is measured, and a timestamp is registered. These data is saved into the HM2 system automatically. Testing compounds, you can get the VS1 to help you do a dose calculation. And these will be stored as well inside the system with the other data. On group housing, so this is where we differ with the HM2 system from other similar systems. We believe that housing animals as close as possible to their natural environment, that we get better and more accurate results due to better welfare. We see that animal welfare bodies admit group housing policies, just like the EU Directive 2010/63 on the protection of animals used for scientific purposes. This roughly states that you cannot isolate social animals for longer periods of time. So if legislation is moving this way, why shouldn't we? Refinement. So the housing the animals in social groups complies with the 3 R's as well. Yeah, and that's on refinement, of course. Minimizing single housing. Sorry. The HM2 system is the home cage. So if you're planning a new experiment on food intake, you don't have any adaptation to new conditions. So you really have a good baseline from the past weeks the animal's been into the system. So you're really ready to go. Using the HM2 system, you get higher throughput. The actual numbers of animals per cage vary from with strain legislation and also experience of each country, state, and facility, yeah, and even the technician. So it's tough to get to give a overall general estimation, but it's very country and specific. But I will say that our typical experience with mice, for example, the male C57 Black 6 is 4 per cage, because these can be rough on each other. Females are often housed by 8 or even more individuals. When we're talking about rats, the lean rats, we typically see 4 animals per cage. Bigger or obese rats up to 800 grams is 3 per cage. Comparing to single or pair housing, you really get a shorter timeline in your project due to habituation and more animals per equipment. So I just wanted a slide about the client server solution as well. So to explain the diagram here, the client server solution spans 3 environments: the animal facility, the office, and the server room. Placing the database on the corporate server makes the IT department responsible for backup and maintenance of the database. So this really secures valuable raw data. Excuse me. The light blue area we see on the presentation here is the animal facility. Over here is the office area, and up here we see the server room. So down at the animal facility we have a 4-station setup, bench top, and over here we see a rack system of 6 stations. These are controlled by a computer, and you can attach your VS1 weigh station to this. What's really important to understand about this system is that each HM2 station has its own internal database. So when data is recorded on this system, it's initially stored locally and then synchronized to a corporate server when network connection is made. So we don't need to worry about network connection breakdowns, because then the data will be stored locally until network is up again. So I tried to make an illustration by animation here, so I'll just run through this. So firstly, we see a scientist sitting in his office planning his experiment, and he will submit his project onto the database on the server. He lets the technician know that the study is ready, so the technician will now assign animals into his project. So this means that these animals cannot be used in other projects than this. So now the technician initiates the study, and meanwhile he might do some body weighing procedures and even dosing projects and submit them as well to the corporate server. So data is stored on the same database. Meanwhile, let's say that 24 hours has passed and the scientist wants to see how his project is going. So he can do this without interfering anything, because he can just run his report and extract the data to his computer and analyze it. And the data will just start continue flowing from his project. His colleague might submit a project as well on other HM2 system. This goes into the same database and will not disturb anything, and the technician can assign animals into this as well, and then data really starts to flow. So I know this is a bit confusing, it looks very stressful, but it's too simple to state that a lot of things going on, but you find the data very centralized to one place. All right. Now a bit about the software, and especially the Cockpit View here. So I won't go into much detail other than that this control room where we monitor and control the system. So we have 3 sections I will highlight here. The first one is the cage overview. So here we can see that 8 stations are connected to the system. These are green. This means they are in running session. 6 other pictograms is possible. So let's say there's a service on one of them, then a different pictogram will show up. But I won't go into that now, just to show this is a really nice way of seeing status on each station. The next one is the live cage view. So here we actually see that cage number 1 here. We see the 8 stations here. So cage number 1 is selected. So here we see a chamber event list. So here we can see if when the sensors are on and off, or if you're doing service on the channels, we have a slider we push in so we can do service. This will be registered here. We have unidentified. I'll come back to that. And we have animal 1 to 4 that are registered into station number 1. Now we can see animal number 1 is selected. So here we see a timestamp of an intake done and the duration. So we see this is actually in milliseconds, so it's very precise. Okay, over here in the graphic is a graphical live view, and also we can see the cage configuration. So here we see food is in channel number 1 and water is in channel number 2. So if one of these animals goes inside these channels, it will actually say animal number 3 is inside the channel. So it's a nice way to see if everything's working well. Okay, moving on to the next view, which is the accumulated data per animal view. So here we see cage 1 to 8. Here we see all the animals per cage. We see the status of each system. We see session duration, we see time remaining, we see the 2 channels, the accumulated intake, number of events, and time duration. And also over here we see time active and time resting. So here we can see that the peer sensor is cage-based, so you see it's the same for all 4 animals. Okay. So the last thing, a nice feature here, we call it a health monitor. You can double click on this table, you get a bar chart, and this tells you also the accumulated data. In this case, the last 24 hours is selected. So here we see the intake. So here we'll also see if any of the animals are not feeding. So in that way, it's a good health monitor. What we also see is that we have a food limit that's yellow. So again here we see that there's a problem in one of the station, maybe a food pellet is stuck between the hopper and the channel, or maybe a technician has been interfering with the study with an animal inside the channel. The last thing about this is you can see a red bar as well. You would see a red bar here. This means that there's an unidentified event going on. So maybe a wrong animal has been placed. An animal has been placed in a wrong cage, or maybe a tag is missing. We don't see that very often, but of course we can't predict the human error. So this is just to tell you that it's a good quality monitor and health monitor as well for the animals. Okay, moving on to the last slide, which is shortly about the results. So when sitting in the cockpit view, we can select results. From here we can configure and generate reports and export them to a preferred data analysis and visualization software. We have made a easy five-step procedure from raw data to graph. And these are done by custom-made reports or predefined. So yeah, you decide on that. To generate a different report, you can come back at any time. So these raw data is stored safely on the database. I have a video of this, but I won't show it here. It's possible to show in the Q&A session if after the presentation, if somebody's interested in this. Mads and Nils will come into more details about the data outcome. So I think I'll leave it from here to Mads Kølby now. So you got the baton now, Mads. Thank you.
Thank you, Renee. I hope you can hear me also in the audience. So I will start talking about some setting up the experiments and walk you through a typical day. And normally when we start setting up experiments, you need to insert this RFID chip. And when we insert it in subcutaneously behind the neck and down on the dorsum of the animal here. And we usually have a healing time when we do this of about 2 days or so in normal mice. And in obese mice, we need a slightly longer healing time. the procedure for inserting this RFID chip is typically, for us, it takes about 3 minutes under isoflurane anesthesia. Other places in the world I've heard about, you can insert these without anesthesia as well. We just have experience with isoflurane, and it takes 3 minutes, and we glue it together, and it works fine. We usually have the mice housed outside the system. The system has these channels out to either the feeding or the water hopper, and this channel is narrow, so it scratches the backs of the mice. So usually we get them to heal somewhere else outside the system for 2 days, 4 days if it's DBDB mice. Of course, you can also, when you have this setup, you also run more experiments immediately, so we usually do the surgery quite some time before we actually use them in the system. When we place them in the system, which you see here on the right, we usually have an habituation period of up to 4 days, but at least 2 days is needed. But we observe the data during this period to see if we get stable readings from their food intake and water intake. So we need to figure out if they can figure out actually to get into the channels and eat and drink. We usually have experience with housing up to 4 to 8 animals per cage, and if it's DBDB mice, DBDB mice have the lack of the leptin receptor in the brain and are never satiate, so that means they eat all the time, and they get more aggressive getting into this feeding food channel if there are too many mice in the cage. So we have personally here a max of 4 mice if it's the DBDB phenotype. Usually also if it's males, they get more aggressive also if you have a house too many and they need to fight over the food getting into the channels here. So you can observe for tail bites or you can find that they get tail bites if you have too many mice housed at the same time. You need also to choose the right size of the feeding channel. It depends on the weight of the animal, how big they are. It needs to be slightly uncomfortable for the mice, otherwise they sit out in the channel all the time, and they shouldn't do that. And then finally, you need to choose the right content in the channel, so the mice in the habituation period are subjected to the settings that you want during your experiment. So either food or food water, which is the most normal setting we have, or 2 bottles for preference. When you begin the experiment, this can last for whatever long time you want to observe them in, because they're housed in the right social context. There are actually no limits, at least where we're working. So the first experiment I'm going to walk you through is the food and water intake over a longer period of time in 2 different mouse strains, and then we can observe the accumulated food intake, meal size, frequency, and so on. Nils is going to walk you through a dosing experiment where we have a leptin test. It's more of an acute test. And then finally he's going to show a 2-bottle preference test and ad lib food on the cage lid. So in the first experiment here we're going to show you today is food and water monitoring over a long period of time in 2 genotypes of animals. We have used the same gender, it's males, so we've housed 4 males together. You cannot house them in new groups if it's males, because they start fighting if you do that, so they have to be already accustomed to each other. We use a day-night cycle of 12 to 12 hours, so light from 6 a.m. to 6 p.m. In this experiment we also used high-fat diets. One drawback with this is that you have to make sure that it falls down in the hopper so it's accessible to the animal, so you need to check every day. If you just use normal chow, this is never the case. You should add only 2 to 3 days of food, because it becomes rancid at room temperature if you have it too long outside. Furthermore, in the software setup we can set them up as 2 groups, genotype 1 and 2. And this group kind of feature you set up with the genotype 1 or 2 can be within or between cages. So this is definitely only assigned to the RFID chip to the mouse. That defines the group. and you can have different groups treatments in the same cage. So just an example from your daily work would be if you have a litter of heterozygotes breedings, you can take all of those with different genotypes, place them in one cage where they're actually used to each other because they're from the same breeding pair, but you can assign them to different genotype groups and still observe how the food and water intake goes. So in this experiment here, we house them actually in different cages to different genotypes. And here we have an observation of up to 336 hours. And you can see that they have a circadian rhythm where they eat more during the dark phase and little during the light phase. What you can also observe is that we did also include the habituation period where they're trying to learn how to use the system. If you use a different genotype, I'm not going to show that here, but if you have a different genotype like the DBDB mice, you would see a more equal consumption. So you'll also have a higher consumption during the day in these mice type here. Furthermore, an observation you can also observe is that the water intake usually follows the food intake. So they drink more during the dark phase where they also usually eat more, and they usually drink more or less the same amount of water as they eat food. If you have any increases of water intake compared to food intake, this can be an indication of nausea or diabetes, loss of fluid, or many other things actually. But usually this is the case that water follows the food intake. So if we zoom in on after the habituation period where we have stable readings on the circadian rhythm, you can see in 15 minutes of resolution here that you can calculate that they eat 82% during the night phase and 18% during the light phase. Furthermore, if you make curves of accumulated food intake, this is actually from the same animals. So you can also have the accumulated food intake you see here in the beginning. There is no difference between the two genotypes because they're still habituating to the system. But when they have habituated, they start diverging the curves between the wild type up here and the genotype X here down at the bottom. You can also do a curve like this or a graph like this where you can take the total food intake accumulated in the whole observation period. This is something you can also do if you do manual recordings of the food intake by just weighing every 12 hours. But of course you lose a lot of data as you saw on the circadian rhythm as shown on the previous slide. Furthermore, you can also see here on the farthest right that after the habituation period you can see that the genotype X is eating less. This is the wild type. They eat more. So this is the difference between the two. What is also nice here from the HM2 system is that n equals 1 is equal to 1 mouse in this system and not 1 cage. If you do a cage-based recording of the food intake, n equal 1 would be the cage and not the amount of mice that you put in the system. Furthermore, the food intake measurements during a manual recording, just by weighing the food, you have food loss like powder down into the cage, which will overestimate the food intake during your experiment. This is not the case in the HM2 system. Just to point out a bit more or specify a bit more what Renee said, there is a hover beneath the channel that picks up all food that is dropped. That's one thing. If there is evaporation from food or the water hover, the spillage system, this is also weighed, so you don't get a measurement error during the actual meal or food water intake. And furthermore, the weight is annulled immediately when the mouse enters the system. So the weight at when the mouse enters the system is recorded, and the weight is recorded when the mouse exits the system. So any evaporation from the spillage has no difference on the recording of the food intake. Furthermore, you can also get kinetics from the food and water intake. Here you can see that the wild type mice eats less meals, but they are of a bigger size compared to the genotype X. The genotype X, as I showed before, eats less, but a higher number of meals, but they are of a smaller size. And deducted from here, you can also calculate the food mean food interval, food intake interval. So that means the time between two meals on average, and that's lower, of course, in genotype X, as indicated over here, because you have a higher number of meals. Furthermore, you can also get a very nice kinetics from the system. So we have a measurement we call the first meal latency. This is what defined from when the dark phase initiates, the mice eats. This typically starts eating. What you can observe here on this curve here is that you can also see the hierarchy in the cage. And what is interesting is that this hierarchy is actually stable across cages. So what I'm showing you here is the alpha male, the second one in the hierarchy, the third and the fourth. That's what you can see up here. But the points here is from one cage, and the point you see here, here, and here are actually from different cages and different populations of mice. So this is pretty stable across genotypes, cages, and mice and time. So that means the alpha mouse usually goes out within 12 minutes, the next one within 30 minutes, and the last one after 75 minutes, and the fourth one, well, he gets foods when the other ones allow him to eat. And here you see the second one goes out in the food hover, and you can see that's after about 30 minutes typically. So using this kinetic, you can observe this across cages, mice, and time. Can we use this for anything? You can actually use this for if you treat the mice in a cage with compounds that alter satiety or causes nausea, you should observe changes in meal size, meal latency, size, time, or frequency. And finally here, you can also use the Weighin Station, as Renee showed you here. So the mice, the wild-type ones that eat more, also accumulate more weight during the experiment, whereas the genotype X do not actually accumulate weight during the observation. And also the peer sensor you observed on the top of the cage can monitor active and resting time, so you can also observe if they're more active during the dark phase, as they should be. And you can also observe that you get a cage activity monitoring. The genotype X here is more active during all the observations that we made, also across several experiments. And this can be due to either that they are hyperactive, or you have they are more awake during the day, or they have a different population within the cage. Some are more active than others at different times of day. So this is just a rough estimate. Here in the middle, just showed you an entirely different procedure we also do, which is an MRI scan for body composition. This is not part of the system, of course, but the problem here is that the mice have RFID chips, and these are not compatible with the HM2 system. So that's a small drawback in the system, HM2 system. So we scanned them before we put in the chip, and we scan them after the chip is extracted. So in summary, we can, if you compare manual versus HM2-based food intake, in normal manual food intake monitoring, you'll have a cage-based food intake, for example, per 12 hours, depending on how good a PhD student you have in your lab, how often they want to go out and measuring the food. You have problems with food lost into the cage in form of powder, which makes you overestimate the amount of food intake that you have. And you get no kinetics data, no meal number, no meal size, no very good resolution or very low resolution, and you get a cage average only. In the HM2 system, you can still house them in social context. You get high resolution and food intake down to 1 minute resolution if you really want to. You can get kinetics on individual mice, and you can also get more data. You stress the mouse less because you don't go up to the cages during the experiment more than once daily. And one drawback is that MRI scans are not possible using the RFID chip. And another drawback is that the activity is cage-based and not individual.
Okay, thank you so much. I'll take you through the last two experiments we've been doing in the HM2 system. The first one is an acute dosing experiment, the leptin test, and just to make sure we're on the same page, I would just quickly mention that leptin is a signal from the adipose tissue to the brain, signaling satiety. So it's a signal on whether you should eat more or not, basically. So high levels of leptin will, sorry, will stop you from eating. So just to make sure we are on the same page. For this experiment, we again used the wild type and genotype X animals. They were microchip tagged and housed in the HM2 system. And prior to the dosing, we kept them for 2 to 3 days to get a stable baseline measurement of their intake. Then we injected them with leptin immediately before the dark phase to see if we could get a suppression of the food intake. And this is sort of a basic test to see if you have an eating phenotype in your animals, if this is leptin dependent or not. And what you do is you analyze the reduction in food intake compared to the baseline of the animals. There's a couple of considerations for doing acute experiments. One I would just quickly mention here is that the system allows you or has the possibility to do a running start. If you do really acute experiments, say you have an ICV cannulation of your animal and you want to do angiotensin II test to induce thirst in the animals, something like that, you can start animals while dosing other animals, so you can get really a really precise start if you have these really acute experiments. Also, when doing dosing, randomization is important for your experiment. Mads gave a nice presentation of the hierarchy in a cage. And say you assign different dose groups to the animals in the system, it's quite important. Say you have a drug that affects first meal latency in the animals, it's quite important that you don't put all the alpha males in one group. But luckily this system allows you to collect this data beforehand and set up your groups so you get the best results possible. Also, it's quite important with dosing experiments to do sham injections, practice with the animals before the actual experiments. What we like to do is just to do this while we collect the baseline data, and you can really see if your handling and injections of the animals disturb the data in any way, and you can see when their intake is stable even though you handle them. So you can see when the animals are ready to dose, basically. So showing you some data from the experiment. You got the food intake on the y-axis here, and on the x-axis the different time periods leading up to the experiment. And you see a nice stable food intake in both genotypes during the dark phase here, as Mads has mentioned before. This is the day of leptin dosing, and you see a nice suppression of the food intake, maybe approximately 60 or 70% reduction in the food intake. So the injection seems to work in both genotypes. Now what you can do here is go further into this data and do a graph like this. Again, we have food intake on the y-axis, and the dots on the left here is individual animals and an average of the baseline food intake during a dark period. The dots on the right are the intake on the day of the leptin test in the same time period. And what this allows you to do is to spot any flaws in your experiment. Looking at the top graph here, you see the red arrow, and this is clearly an animal not responding to the drug at all. So what this allows me to do is to go in, maybe this was not injected right, and I can go check that with an ELISA in the plasma or something. It allows me to check really if all is well here. Going to the other genotype, you again see we have a non-responsive animal here at the top, but also that we have two animals not eating at all here. They're basically zero in food intake during the entire period. This might lead me to suspect that these animals are not well, and I could perhaps go to the room and check for any blood in the cage, or see if they are in any way feeling malaise. I can also maybe check the activity in the cage and see if it's down, if they're not moving around at all. A different way to show the results, it's accumulated time curves like these. Again, food intake and time on the x-axis here, and here you can go for any resolution you would like, really. What I've done here is 15 minutes intervals for the first 3 hours of the test, and I've only shown you the wild type animals to make it more clear, but you see a nice reduction in the food intake, very little variation here. If we go to a 1 hour resolution instead, again you see this nice reduction in the beginning, but looking from 8 hours and beyond here, you really see you start getting a lot of variation in the data out here in the dosed animals. And what you can do is go further into this by again doing individual curves for each animal, which I've done here. And this allows you to really dissect what's going on after your dosing. You see again, this is the green curve here on top with the black arrow on top is probably the non-responsive animal, just eating like the wild type ones. But you can also dissect out other outliers like the 2 arrows on the bottom here. One is responding to the drug, but then perhaps it's a fast metabolizer, at least the effect stops abruptly after 10 hours and it catches up. The other one is sort of responding intermediately here during the entire period. So you can get clues from data like this on the efficacy of your drug or the time your drug is active and so on, if there's a difference. Also, if there's a difference between your genotypes here, you could do the same plot for your genotype X animals. So these were some consideration when doing dosing tests. The other test I would like to share with you is an alcohol preference test we did in the system. So this is going into a bit more behavioral studies. And what we do here is set up both channels in the system for bottles, and we keep ad-lib food in the cage lid so the animals can eat from there at any time. We run for 3 days with water in each bottle to ensure there's no preference for any of the bottles, and then we switch and introduce the ethanol. Then we switch the bottles every 3 days to make sure there's no bias, and we repeat this 4 times. So that's the basic protocol for the experiment. Some of the consideration for this type of experiment is that you would like to conserve the water in some way. We like to acidify the water with citric acid to a pH around 3. This will not affect the animals in any way, but it will make sure you have no bacterial growth in your bottles. Also, it's of course important to clean the system extensively before doing experiments like this. Also, the system, we like to use the log in the system to report anything you do in the room to make sure you really log everything for this type of experiment. This is some of the data, and this is data for the first 3 days with water in both bottles. And we run this just to make sure, as I said, that they like each bottle the same. On the left here you see a graph from one of our cages, and really this is what we like to see. The bottom graph is the water intake from channel 1, and the top graph is both channels accumulated. So basically the gray area is one bottle, the brown area is the other bottle here. And what we see on the graph on the left here is a nice distribution between both bottles. We had trouble with another cage here, cage 3, and we did the same, and what you can easily observe here is that they're really only using one of the bottles. They're only drinking from channel 1. So at the time with the arrow here, we switched the bottles, and you can clearly appreciate they stopped drinking from one bottle and switched to the other one. So it's not a place preference in the room, but perhaps we have infection in one of the bottles, or at least some taste perhaps associated with the water that they don't like. So they really go for a specific bottle here, which is of course not good if you want to test a preference. Moving on, after solving the problem, we collected the data, and this is the general compiled data from the experiment. The y-axis here shows the alcohol preference percent, which is just the consumed amount of 10% alcohol divided by the total liquid intake. And this would seem like genotype X prefers alcohol much more than our wild-type mice. But what this system allows you to do is to delve a bit more into these data. And like I mentioned in the protocol, what we did here was switch the bottles every 3 days. And this is a graph showing each of these 4 periods and each of the days during that period. So basically each column here is a successive day in the experiment, and every time the number on the x-axis goes up, we switch the bottles. So what you can appreciate on the right here for the wild-type animals, the ethanol bottle is really interesting once we introduce it, and they drink a lot, but quickly they settle on their preference here around 20%. After the first switch, they have to relearn that we switched the bottles, but they quickly pick up the system, and really after 5 days they end up on their alcohol preference here around 20%. So the reason we saw much higher alcohol consumption in the other genotype is perhaps not that they prefer alcohol, but perhaps that they are a bit slower learners. As you can see here, it takes a longer time to get to the level here, but actually during the 4th period here, they're really trying to reach the same level as the wild-type animals. So what you could perhaps get here is a false positive for a different alcohol preference, when really the animals are instead slow learners, perhaps a bit dumb. So really this raised some new questions for us that we can go into, but yeah, certainly this is some exciting stuff you can get from experiments like this. What you could do here is, of course, go further into the data and extract kinetics and so on, like Mads showed you earlier, which would really help you pinpoint what goes on in these strange animals. So to sum up, we have tried to review 3 different types of experiments, a long period of monitoring, acute dosing experiments, and these preference experiments. We tried to highlight pitfalls in surgery and in experiments of this sort, and we tried to highlight the strength of doing these kind of studies in a group-housed environment and in this kind of system. And with this, I will end, and I think we have a Q&A session, so I will hand it back to Martin.
Let's start, Mads, perhaps with you. You had presented a slide where you showed 4 to 8 animals in a cage, but the question here is, what is the maximum number of animals that one can put in a cage to study group-housed ingestive behavior?
That's a very good question, Martin. The system, as I have been told by Renee, is that it has no limit at all. So you can fill it up. But I would say that the limit is depending on genotype, how much food they would actually need to eat. But my experience is 4 to 6 males at maximum and up to 8 females in the cage. So that's in my experience. If I go much higher, I start getting tail bites, and that's an indication of too high amounts of animals in the system.
Okay. Renee, do you have anything to add to that from, based on your experience?
Yeah, I agree with Mads. It's very specific for each country as well regarding legislation. So it's both on the strain, the genotype, and the legislation, and also the experience of using the system. So yeah, but I totally agree with Mads on this.
Okay. How does one know that the system is working properly once it's been set up, and what should users be looking for to ensure proper operation? Renee, why don't we start with you?
Yeah, okay, so we have quite a few health monitoring systems in this in the system. So the first thing we recommend doing when setting up the system is doing a hardware diagnostic. So this helps you. This is a guided check of all detectors. So these results are logged into the system. So so along with other quality indicators like in the chamber event list and online, we have the online condition of the cages, as I showed you on the Cockpit View. We have the meal size limit. We also have unidentified meals and drink, and as well as body weight in and decreases. These things are highlighted in the system, so you'll be aware of if something is wrong, and all these data is logged into the system, so you can actually do a quality report on this to put along with your session results if you want to.
Okay, perfect. And Niels and Mads, maybe along the same lines, I know you mentioned it, but maybe you can just elaborate. How can a user be sure that individual water and food intake is accurate given that there's going to be spillage and evaporation? Can you just sort of maybe repeat and highlight what you had said during your presentation?
Yes. First of all, it seems in the system it logs the weight of the food or the water hover at the time the mouse enters this channel. So any evaporation, for example, from the water spillage tray, is annulled because the weight is weighed, or all the water is weighed at the exact time the mouse goes into the system. So any evaporation would be no problem at all.
Okay. Perfect. Can you comment or elaborate, I guess, again, maybe we'll start with you, Mads and or Niels. Can you comment or elaborate on the use of a system like this for drug administration studies? Somebody has asked if you can, dose an animal orally by putting a compound in the water.
Yes, actually the system would be perfect for that, because what you get by dosing in the water is that you, by following the water intake, you get the precise dose in each animal, and also you get the dosing with a timestamp because you know exactly when they have been drinking. So it allows you to follow the administration of the drug really precisely. So the system would be great for a study like that.
Okay, perfect. A question here, we actually have two questions and perhaps they're related. Do researchers set a predetermined meal size and intermeal interval and then generate meal number and pattern? And then another question from one of our audience members is it possible to control food and water access? I think the question is one of the same. So Niels and Mads, do you want to take a stab at answering that question?
Yes, I can try. The system observes only the amount of food and water that is ingested at any given time. You are not able to control meal size or access in the system to food or water, for example, to shut it down for an hour. If you want to shut down food and water intake for all mice, you will have to close it manually.
Okay.
You can, however, you can set limits for what is a meal, what is the maximum weight of a meal for each mouse. You cannot stop it eating, but you can look, when you look at the data, and you can extract these things. But as Mads says, if you want to restrict the access, you close this metal plate to the channels manually. But this can be done quickly. So it's not.
Okay. That makes sense. Renee, do you have anything to add to the answer that Niels and Mads has just provided?
No, I think that's very, very nice explained, because it's the metal slider. Yeah, then you'll have to do it manually to close the channels. So of course that's a possibility. As Mads was talking about, if you have a good PhD student, then these could do these procedures.
Okay. Okay. Very good. Eveline has asked if the question is, can you refill the food and drink dispensers without interrupting the session? Renee, I'll let you answer that question.
Okay, yes, that's a very good question, and the answer is yes, you can, because the only thing you need to ensure is that there's no animals inside the channel. So what we recommend doing is closing this we talked about this metal slider. So you close the channel off and secures there's no animals inside the channels. Then you can fill up the food hopper and change the water bottle maybe. So as long as there are no animals registered in the channel, you can do exactly this.
Okay, fantastic. A question here from Thomas in the United States is, how does the system handle animals that hoard food? Mads and Nils, may I start with you? Is that a question that you can answer?
Yeah, sure. It's not possible simply for the animals to do this. The design is that they can't really take a portion of food and bring it back to the cage, because this would destroy the measurements really. So they're not able to do that. If you keep ad-lib food in the cage and measure with 2 bottles, for instance, then of course they will be able to do it through the cage lid, but not if you monitor the food intake in the channels. Perhaps Renee can back me up here.
Yeah, and in the food hopper we have bars that are fairly close, so they can't get large sizes of pellets out of the hopper, and also there's a grid under in the channel, so any spillage will drop into the tray under. So it's not a common problem we see. So.
Okay. Very good. And Nils, I think this question came in to you specifically while you were presenting your slides on the leptin test. The question from Ramon is, how can you tell when the mice are ready for injections? I think you may have mentioned something about that, and that point was missed. Can you just repeat that?
One point is that we like to keep them in the system until they have a stable baseline intake. So when we don't interfere, they have to have a stable intake, so we really have something to compare after dosing. The other thing is that we like to sham inject the animals, like just poke them with a needle or inject saline or something inert to get them used to the procedure. Now, if they are stressed out because of this handling, we might see it in the data. If we don't, then of course the animals should be ready, as they are not stressed out by our handling and so on. So that's sort of a built-in check that you have done enough handling of the animals so that they are ready.
Okay, great. Renee, is it possible to add video cameras to a system like this so that one can record social interaction between animals?
Okay, yes, we actually have a possibility to do this, to add a webcam to manually monitor the animals. So it has nothing to do with phenotyping or any other type of software combined with it at this point. But yes, we can fit a video camera on a webcam on the system to study behavior in general. Yes.
Okay. I may make this our last question. Again, we do have many, and it's almost 10 after the hour. But a couple of people have asked how data is extracted and how quickly it can be analyzed. Renee, you mentioned that you have a slide that demonstrates this functionality. So what I'm going to do is I'm going to pass controls back to you. And maybe you can just give us a brief demonstration of how that what that would look like with your system.
Yes. So hopefully you see the slide here.
Yes. Yes.
Yes. Good. So we have a study here with 40 rats in 5 dose groups, and please note that three animals were taken out. So the group setting in animal numbers would be 78877. So this means that we need to put in empty columns. So the system automatically does this. This example is a 24-hour food intake study over 5 days. So yeah, and I will try to take you through this 5-step process. This is a video clip, so I will try to pause the video and just comment on the different settings as we are running through it. So firstly, we need to select the experiment, the study, and the session you want to extract data from. So hopefully you see it on your screen. Hopefully you have a big screen because the text isn't that big. Yeah. So now we have the the session selected. Now we need to select the time slot and duration for this. So this is done over here. So I won't go into details at this point, but it was just to show you how quickly we can do this and how easy it actually is. So now we execute and isolate the data. So here we select the protocol, in this case the 24-hour intake. So here we also see other protocols that are predefined, and of course you can do your own custom-made protocol, so it's very easy to select and then just move on. So now data will be analyzed of these 37 rats. So it's a lot of data in 5 days of the intake. So it should be ready right about now. So here we see the data. So now we need to sort them by groups, and this is for preparing them for the analyzing software. So now we see we have sorted them by groups and selected vertical mode. So here we actually see on the first group that we have an empty column because there's only 7 animals in this group. So number 8 is just a clear field. So you select clipboard and 1-day interval, and then you can paste it into your analyzing and visualization software. In this case, it's GraphPad Prism. Can be any other program that you're using. So then you have the graph. So you can do this in 1 minute if you, of course, you need the routine, and you have to have the pre-prepared reports and the analyzing software as well. So I hope it makes sense.
Perfect. And actually, it does seem like a lot of data that is extracted very fast. Can you just remind everyone, particularly what parameters does one extract? So meal size, what else typically is organized into those columns?
Sorry, I don't follow much.
Right. So in your presentation, your last slide had a list of the parameters that one would extract from the data.
Yeah. Yes. So it's typically it's meal size, and meals, and you can go into bouts as well. It's a bit more complicated, but it's possible. You get the intermeal interval as well.